Friends, welcome back to Biotech Simplified. Today, we are going to discuss about types of regeneration. Earlier, I have put a video on what is regeneration. I hope you have watched that before going on to the types of regeneration. So, the most commonly seen type of regenerations are stem cell mediated, epimorphosis, morphallaxis, and compensatory. So now let's move to the first type of regeneration, which is stem cell mediated regeneration. The most common example of stem cell mediated regeneration is seen in hair. That is, we all know that once hair is cut, the hair regrows from the hair follicle. So this is a type of regeneration, and this type is known as stem cell mediated regeneration. So now let's move to the next type of regeneration, which is compensatory regeneration. We all know that in human beings, the ability or the capacity of regeneration is much low. And the most commonly seen regeneration in human beings is compensatory regeneration. In humans, if a part is lost, then the size increases so that it replaces the gap. And this is usually seen in liver, and liver is the best example of compensatory regeneration. In this video, I would like to discuss more on epimorphosis and morphallaxis. So let's discuss each in detail in the upcoming slides. Morphosis. Epimorphosis is a type of regeneration generally seen in amphibians as well as wall lizards' limbs. That is, if a limb is lost, the regeneration pattern seen there is epimorphosis. Now, let us discuss what is the mechanism involved in epimorphosis. So, the first mechanism or the first step in epimorphosis is de-differentiation. A reprogramming pattern is seen in epimorphosis. That is, the existing adult cell it de-differentiate so that an undifferentiated mass of cell is formed there. From this undifferentiated mass of cell, a pluripotent state is developed and redifferentiation occurs. And this is how they restore the missing cell or the organ. As I discussed in the earlier slide, the epimorphosis is generally seen in amphibians and salamanders' limbs. So it is a limb regeneration technique. After the limb is being lost or the limb is being cut, a wound epidermis develops there, and this is the first step. And after that, a blastema develops there, and it is from this blastema the limb is being regenerated. So now let's understand the mechanism from a more detailed diagram. So as I discussed, once the limb is being lost, a healing occurs and wound epidermis develops there. And after that, a de-differentiation occurs and blastema cells appears there. And this region is known as apical epidermal cap. After de-differentiation, undifferentiated cells appears there and the blastema cells develops a pluripotent capability. It also requires nerve cells so that a regeneration happens there. It apart from the nerve cells, it also requires retinoic acids, which is needed for the axis development, and FGF8. Redifferentiation occurs and the lost parts is being restored. You understood what is epimorphosis. Now let's discuss about what is morphallaxis. In this regeneration, the missing organ is restored by cellular organization, repatterning, and remodeling. The most common example of morphallaxis is Hydra. You must be familiar about the structure of a Hydra. A Hydra has a head, body, and a foot. The head region has tentacles as well as a hypostome. Let's discuss the different cases of Hydra regeneration. The first case is when the hypostome is cut and placed in the trunk area of another Hydra. A secondary axis develops in the Hydra, that is, a head region develops along with the Hydra. Second case is when from the food, that is from the basal disc is cut and it is placed in another hydra. In this case, a food develops along with the parent hydra. That is, a secondary axis is seen here too, but it is a food. So the difference between case 1 and case 2 is that when the hypostome is removed and placed in the trunk, a head is developed, and whereas when the foot region is cut and placed in the trunk region, a foot is developed along with the parent hydra. I hope these two cases are clear, and if this is clear, let's move to the next cases. Previous slide I explained what will happen when a hypostome is cut and placed in the trunk region. That is, in this case, a secondary axis will develop along with the parent axis. So, what might happen if the entire region is cut and placed in the trunk region? So, in this case, a secondary axis was not observed. And the second last case is when the head region is cut and placed into the trunk region of another hydra where the head is being removed. In this case, a secondary axis is being observed. And the last case is when the head is cut and placed into the foot region of another hydra, that is, a hydra in which head is present. In this case, a secondary axis is being seen, that is, a head is seen along with the parent axis. What might be the conclusions obtained from these cases? The first conclusion is that there is some substance present in the head which leads to secondary axis development, that is, which leads to head development along with the parent axis. Similarly, there is some substance present in the foot which leads to secondary axis along with the parent axis. So the region with the substance present in the head is termed as head activating gradient, and the substance present in the foot is termed as foot activating gradient. And these two gradients inhibit each other, that is, the head activating gradient inhibit the foot activating gradient, similarly vice versa. The second conclusion obtained from these is that along with the head activating gradient, there is another gradient present in the head region, which is head inhibiting gradient. The head inhibiting gradient is maximum when it comes to the head region, and it is least when it comes to the foot region. And it was because of this reason, as we saw in the earlier case, when the head is removed and placed in the trunk, a head or the secondary axis is not developed. And this is because it is in close proximity with the head, so it inhibits the head. So even though head activating gradient is present, because of the presence of head inhibiting gradient, there is no secondary axis. But in the case when the head is removed and placed in the foot region, and a secondary axis or the head is developed in the foot region, and this is because the head activating gradient is maximum towards the head, whereas it's least when it comes to the foot region. Let's summarize what we learned in this video. First is epimorphosis. Epimorphosis is a limb regeneration technique. When a limb is lost, dedifferentiation occurs and blastema develops. After that, redifferentiation occurs, and this is how restoration happens. Second technique is morphallaxis. Morphallaxis is seen in Hydra. In Hydra, due to the presence of head activating gradient as well as the presence of foot activating gradient, restore or regeneration happens. Hope this video was useful for you. For more such content, please follow Biotech Simplified. Please do like, share and subscribe to my channel.